Hello everybody, I'm here to introduce to you the shiny app in our package, VIEWpoly. This tool is part of the Tools for Genomics-Assisted Breeding and Polyploids project. You can find more information about this project, the other tools, the participants, and events on this website. Access the website address in the description of this video. The VIEWpoly is hosted in CRAN. You can install the package using the install packages command. The VIEWpoly development version is hosted in GitHub. To install the development version package, you will need the DevTools package installed and use its function install GitHub with the VIEWpoly repository address. If your operating system is Windows, you may need to install our tools before installing DevTools. Once installed, you can open the application using the command run app. You can use RStudio Viewer or open the app in your browser. The app is organized into 4 tabs, the Input Data tab, and 3 modules, ViewQTL, ViewGenome, and ViewMap. Click in the top menu to access each one. In the Input Data tab, you can upload the dataset to be displayed in the next tabs. Included in the package is a default dataset that serves as an example for the app features. The example dataset derives from a tetraploid potato mapping population linkage map, QTL analysis, and genomic information. To save space in the package repository, it contains just a subset of the individuals. However, there are complete example datasets available that you can download accessing this link, which also includes data from a hexaploid sweetpotato mapping population. You can upload files of linkage maps, QTL analysis, and/or genomic information. If you only submit linkage map files, only features related to it will be displayed by VIEWpoly. The same happens with QTL analysis. But for the genomic browser features, besides the genomic information, at least one of the others is required to access the genome browser features. Click the plus symbol on the right side of the Upload Linkage Map Files box to open the options for uploading the linkage map information. Once you open one of them, you can see instructions about how the file should be uploaded. In the example below, it requires an RData file containing an R object of class mappoly.map. The first provided link points to the MapPoly tutorial, where you can find further information about all the procedures to build a linkage map. The second points to the VIEWpoly tutorial that contains an example code of how to obtain this input. Click on Browse to select the file on your computer and upload it into the app. Wait for the upload to complete and click on the Submit button. In this case, Submit MapPoly. After clicking Submit, if you want to change any of the uploaded files, you must click on the Reset button before uploading new files. If you choose to submit polymapper files, you should do the same procedure. But remember that VIEWpoly only evaluates one dataset at a time. If you upload MapPoly and polymapper, only the last files uploaded will be evaluated. If you choose to submit the linkage map files as text, TSV, or CSV files, you can download example files as reference when formatting your data. You can do the same process with QTL analysis files. Upload files for one of the 4 options: QTL Poly, DiaQTL, PolyQTLR, or standard formats. From the QTL analysis, at least 4 files are required. Just as before, the first provided link points to the QTL analysis software tutorial, where you can find further information about all of the procedures to build a linkage map. The second points to the VIEWpoly tutorial that contains an example code of how to obtain this input. After loading all the required files, click on Submit. There are also example files available to use as references for uploading standard files. By providing URLs or uploading files in this section, you will have access to the Genome Tab Features. All information will be included as tracks into the JBrowser Genome Explorer. Only the assembly FASTA file is required to access the feature, while other genomic data, such as gene annotation and alignment files, are optional. You just need to make sure that linkage, QTL, and genome-related files are tied to each other. For example, uploaded QTL results were obtained using the uploaded linkage map, and both were based on the same uploaded genome assembly and gene annotation versions. Please only choose one of the options for all submitted files, URL or uploaded files. Do not submit some of them through upload and others through URL. Providing the files through URL improves VIEWpoly efficiency. You will not only need the files, but their respective index as well. In the VIEWpoly tutorial, the authors show a few ways of obtaining them. You should upload them together to the app. Once you submitted the linkage map and QTL analysis results, VIEWpoly converts all files to a single R object of class VIEWpoly. It keeps only the information required for building the app graphics for QTL and linkage map analysis. You can set the name of the dataset R object using the box. Avoid using special characters or spaces. Click on Download to save as an R data object. Once you have a VIEWpoly object file, you can access VIEWpoly features for this dataset just submitting the single file in the Upload VIEWpoly dataset section. You can also load your VIEWpoly dataset in your R environment, and it will be listed in this section. If you have more than one VIEWpoly dataset available in your R environment, all of them will be listed. If you uploaded the QTL analysis information for any of the software options, you can access the features in the QTL tab. These graphics and information may change according to the software used to perform the QTL analysis. You can explore any of the linkage groups or the phenotypes together or separately. Select them using the two upper boxes. Once you selected the group and phenotypes, the QTL profile curve will be plotted. On the y-axis, it shows a measure of statistical significance for each trait. This can be different according to the software used. The x-axis is relative to the linkage map position. The triangles at the bottom represent the peak of significant QTL. The black line spanning the triangle defines the QTL supporting interval. You can download this figure and all other figures from VIEWpoly, selecting the format in the File Type field and pressing the Download button just on top of each figure. For further information about these graphics, please check QTL Poly, DiaQTL, and PolyMapper tutorials and papers. We include links for them in this video description. You can click and drag your mouse cursor to select the triangles at the bottom of the graphic to explore particular QTL effects, progeny haplotypes, breeding values, and to get the QTL summary table in the next sections. Click on the plus in the right corner of the Effects section to see the graphics. By default, VIEWpoly displays the bar design. It displays the additive effect for each of the parent's alleles on the selected QTL positions. Colors emphasize the intensity of the effects. Changing the design to additive circle, the intensity of the effects is plotted in a circular graph. The selected QTL from the same linkage group are plotted together in the same graphic. To be able to compare the effects across different QTL and phenotypes, the values are standardized to be between -1, the center of the circle, and 1. The dot color is more intense close to the extreme values, -1 and 1, and the transparency increases when they're close to 0. Changing the design to alleles combination, a heat map is plotted with the combined alleles additive effect sum in the bottom diagonal. The upper diagonal shows the diagenic effects which can only be estimated in DiaQTL software. This design is only available for QTL Poly and DiaQTL software results. Now you can search the individuals in progeny that have specific alleles. To access this feature, first set the design to additive bar. Click on the Update Available Haplotypes button, and then on the Select Haplotypes gray box to present all possible parents' alleles for the QTL selected in the QTL profile graphic. You can select any combination of alleles. For example, I will pick here the allele P1.1 and P2.1 in linkage group 5, position 0, referring to the QTL of trait ST08, and P1.2 in linkage group 5, position 28, referring to the QTL for trait N108. After clicking on Submit Selected Haplotypes, VIEWpoly will search the individuals in the progeny that have a probability higher than 0.5 of having all the alleles selected. VIEWpoly displays the genotype probability profile for each individual identified with the selected alleles. The QTL position are indicated by dashed vertical lines. If only one linkage group is selected, the graphic will split the probabilities for each possible homologue vertically. If we include the QTL for trait NSO6, which is in chromosome 1, and also try to find individuals that have the allele P1.1 in this QTL position, the graphic design changes to a stacked version because two groups must be represented in this case. If you upload QTL poly results, you can also access the individual's breeding values. Only the effects of the selected QTL in the QTL profile graphic are considered to estimate the breeding values. Here, you can also access a table with a QTL information summary. Let's go to the next tab, the View Genome module. The goal of this module is to relate the QTL position in the linkage map with the available genomic information. The visualization here is made by defining the map range by chromosome. Therefore, you can select only one chromosome to be visualized at a time. You can also select the phenotypes. Then the QTL position and confidence interval are plotted just below the range. Click on the plus in the right corner of the QTL profile section to access a plotly graphic with the QTL significance profile. A scatter plot of the genetic versus the physical positions is displayed to highlight the relationship between the genetic map, the reference genome, the QTL positions and intervals, and the recombination rate along the chromosomes. If you updated the genome information in the Input Data tab, you can click on the Open JBrowser to visualize the genome and all the tracks according to the files uploaded. The range in the genome is set from the position of the first marker to the position of the last marker in the selected linkage map range. You can also explore all of the JBrowser features by clicking on the buttons inside the window. We can, for example, zoom in. Access the menu by clicking in the upper left corner. We included the link for the JBrowser tutorial in the description so you can explore more features. VIEWpoly creates a local server to host the genome files in JBrowser to be able to access it. Then, before exiting the app or changing the genome file, you must turn off the generated server by clicking on the button Local Server. If not, the server will only be off when you close or restart R. If you uploaded the JFF3 file in the Input Data section, a table will be available with annotation information inside the range selected in the linkage map. Next is the View Map module. This module's goal is to provide tools to explore the linkage map properties. You select the phenotypes and the linkage groups in the same way as we did in the last module. The QTL profile graphic is also displayed here. In the map section, the parents' haplotypes and linkage map are plotted related to the selected map range. If the reference and the alternative alleles information are present in the uploaded linkage map files, the variants are presented with different colors for each nucleotide or indel. If this information is not present, red and white will differentiate the biallelic sites. The dot position and color represent the dosage number. The parent's haplotype information used to build the graphic can be downloaded in this section in table format. VIEWpoly also provides a table with the linkage map main characteristics and the linkage map figure. For now, in the VIEWpoly 0.1.0 version, that's it. The authors intend to expand the app to support inputs from other softwares and to follow their updates. If you have any issues, questions, or suggestions about VIEWpoly, you can submit an issue on the GitHub page or send an email to the authors. Access the addresses on the tutorial page. Thank you!